Okay, welcome everyone. I am Ashanti Edwards, the Director for Professional Development at the American Society for Cell Biology. We are pleased to offer Online with LSC as part of ASCB's Professional Development Webinar Series. Today, our facilitator, Cynthia Brand, will be introducing our featured author for this installment of Online with LSC. Cynthia, you can turn on your camera now.
Hello. I want to welcome everyone to this session of Online with LSE. You'll remember that the goal of this webinar series is to be sort of a virtual journal club that can provide a sort of behind the scenes insights into education studies. Okay. So I'm very excited to get to talk to our series of authors today. It's a fascinating study and a really useful annotation of it within the Anatomy of a Research Study feature. So I want to encourage you to submit questions as we go. Please use the Q&A feature. We will be reading those questions, synthesizing those questions, and making them visible as we go. So we really want this to be a conversation as much as possible. Okay. So I'm going to introduce our four authors, and then they will introduce themselves and give a brief overview of their work. So would you all please, as I introduce you, turn on your camera so the audience can sort of see you live. We're going to start with Jennifer Jo Thompson. Jen is an associate research professor in the Department of Crop and Soil Sciences at the University of Georgia. She's director of the Sustainable Food Systems Initiative. Her research interest in science identity and capital is currently focused on efforts to expand equity and participation in the context of agriculture and food systems education. And this is a way to address inequities in our food system. Okay, we also have Danielle Jensen-Ryan, who is faculty in the School of Math and Sciences at Laramie County Community College. Danielle's research explores inclusivity and undergraduate research experiences, particularly for minority, first generation, low income, and women in STEM. Danielle also directs the Human and Public Services Pathway at her college, which supports and connects students to engage in paid internships and community development efforts. Then we have Becca Price, who is professor in the School of Interdisciplinary Arts and Sciences at the University of Washington, Bothell. She's a member of the editorial board of LSE and co-editor of the Anatomy of an Education feature. Her research explores evolution education, inclusivity, and professional development. Becca also directs the Science Teaching Experience program at the University of Washington, which supports graduate students and postdocs as they learn and implement inclusive evidence-based teaching practices. And then finally, we have Clark Coffman, Associate Professor in Genetics, Development, and Cell Biology at Iowa State University. He is the Faculty Fellow for Graduate and Postdoctoral Professional Development in the ISU Center for Excellence in Learning and Teaching. He's on the editorial board of LSE, and he's co-editor of the Anatomy of an Education feature with Becca. Okay. All right, so we look forward to hearing an overview of your work.
Well, thank you very much, Cynthia, for that introduction, and to all of you for attending our webinar. We've had such a wonderful collaboration with CBE Life Sciences Education, as well as during the annotation of our article with Dr. Price and Dr. Coffman, and we feel so fortunate to be able to discuss our work with you today. And so I'm just going to dive right in and provide the context of our original research article that Dr. Thompson and I collaborated on, which is entitled Becoming a Science Person, Faculty Recognition, and the Development of Cultural Capital in the Context of Undergraduate Biology Research. Dr. Jen Thompson is going to follow with more context on our paper after I provide just a touch up front. And then Dr. Coffman and Dr. Price will discuss their annotation of our article following that. So what's the context of our research? Well, our research is built upon previous work that outlines the benefits of students engaging in undergraduate research experiences, particularly how research experiences help students build a science identity. And so you're probably wondering, well, what is a science identity? Well, it's first off an internal sense of oneself as a scientist or a science person, or it's a recognition of yourself by a meaningful other. So this could be a faculty member, a mentor, a peer. And why this is important is because it's a good predictor of whether or not you'll actually persist in STEM if you develop this science identity, particularly for students. So our article focused on the experiences of students participating in undergraduate research experiences, and particularly how their participation was or was not recognized by faculty mentors. And so with this work, we ultimately built on the literature to theorize and operationalize undergraduates' recognition by those important, meaningful others, by that faculty mentor. And so what was our methodology? Well, this is part of a longitudinal study with a biology research network, starting all the way back in 2011 to the present day. So I'm still involved with this biology research network and study the scientific and student learning outcomes associated with undergraduate research. This biology research network started as 5 faculty members at 3 institutions. And as of right now, it's 23 faculty who are supervising different undergraduate students at 21 universities and institutions. And in addition to that, 200 students have participated in the lab experiences, with over 1,000 actually participating in the course-based undergraduate research experiences. So it's a wonderful biology research network. We feel so fortunate to be able to work with them. But our methodology is we conducted student and faculty interviews, okay? So we interviewed 20 students from 10 different institutions, and we interviewed all of the PIs. We also looked to all of our past research interviews that we conducted with students and faculty to really round out our data. What we did was transcribe these interviews, check them for accuracy. Then we uploaded them into Atlas TI, which is our qualitative data management tool, where we coded for themes. So here's the important kicker, right? We systematically compared student data with faculty data. So we compared how students self-recognize themselves and compared that with how faculty recognize them. And what this ultimately led to was us to identify areas of alignment. And most importantly here, what we found was areas of misalignment between students' self-perception and their faculty recognition of their science identity. And so with that very brief context for the beginning, I'm going to now pass this over to Dr. Jen Thompson to go over the results of our study and the main takeaways of our research.
Thank you, Danielle. So what we ended up doing here is through that kind of systematic analysis and comparison of our student data, again, 20 students, with our faculty data, we ended up identifying both, starting with some of the literature around science capital, and also looking in our interviews for ideas that maybe were not already in the literature from the past. We identified a number of domains of cultural capital. And this idea, and I think we're going to talk about this a little bit later, but this idea comes from some the notion that cultural capital is the way that we kind of know how to move through the world with ease within a particular field or domain. So we don't really argue that it's better or worse to have it, but it definitely makes your movement in a field easier. It's kind of like knowing the rules of soccer, knowing how to play, having practiced it, and then being able to get on the field and play some pickup because you know the game. So our goal then was in this process, kind of identifying these places that students talked about what they felt was important for their identity, and finding these areas where faculty also recognized them. So we identified a number of domains of this kind of science-related cultural capital. And in doing that, we also ended up defining what it meant for students to sort of excel in that or have a lot of it, or for students to have very little of it. And again, I just want to really highlight that our objective there wasn't to highlight anything that we considered a deficit among students. But in fact, cultural capital is something that Pierre Bourdieu, a French theorist, says, is learned at the mother's knee. It's something that we develop without really realizing it through a lot of exposure over time. And so the fact that someone doesn't have it doesn't mean that they aren't capable of having it. It means oftentimes they haven't been exposed to and haven't had the opportunity to practice it. So what we tried to do then is look at these domains, identify where our students kind of fell on these continuum across these domains, and tried to use that as an opportunity to then predict or examine the way that their own capital and their own self-recognition aligned, or as Danielle said, misaligned with faculty recognition. And one thing I want to point out here is that in our data and in the paper we wrote, we opted to use a series of vignettes to illustrate this. And so what that means is that even though we interviewed 20 students and we analyzed the data from all 20 students, we selected 5 examples from real students to help illustrate the complexity of capital and the complexity of their life experience and their relationships to faculty mentors. And this was, and again, these are real students, they aren't composites. And the idea here is to also show that these domains of capital are not distributed in a kind of linear way, but to show the complexity. And this allows us to tell their story rather than a lot of qualitative research often gives a number of kind of disembodied quotes. And we felt that because of our focus on science identity, that this would allow us to tell students lived experience and their whole stories, or at least the science identity components of their story. Okay, so I want to say one other thing related to this. I want to dig into one example of this. So one example, one domain, for example, is interest in science. And what we found as we looked across all of our interviews is that some students had this kind of lower, what we described as a kind of lower amount of capital in this world that fell into a place where they were just sort of sort of so-so about science. This one student said, I wasn't really into anything in high school. I didn't really enjoy it at all. I didn't want to go to college. Whereas on the other end of the spectrum, we had students who described things like a particular interest in exploration, in discovery, right? And that constituted this high capital, with this middle domain kind of being excited by a teacher or a course that stimulated their excitement. So we described each of these domains with a kind of low, middle, high based on our data. And then we categorized our participants in this way and then used that to match them up with faculty. And ultimately in this paper, our objective, of course, was to then identify some take-home messages or some take-homes for faculty. And our key take-home that I really want to emphasize is that students who come into science with quote unquote lower capital are absolutely able to learn and develop this capital. But the hitch is they need recognition from faculty. They need to be acknowledged as budding scientists, and they need to be then encouraged to develop that identity so that they then have opportunities, more exposure, more opportunities to practice that identity and to engage within the community of science. So our take-home message is that we really are trying to develop, to encourage faculty and of course institutions to support faculty to broaden that scope of recognition for students. I'm going to turn it over to Becca.
Yeah.
All right.
Thanks, Jen. So I want to just jump in and show you what Jen and Danielle's paper looks like in the Anatomy of an Education Study feature. And I want to show you, first of all, that there are a number of learning lenses that we use. So we're making annotations related to the background, to definitions of certain definitions, instructional practice, research design, and writing tips. So you can click on any of these highlighted areas and see what we've written. So we chose this section of the paper because there are lots of highlights from different lenses, and we're going to focus in on the instructional tip. Okay, so now I'm only highlighting instructional tips in the manuscript, and then I've clicked on this highlighting and I can see, oh, okay, so when we look at this grand conclusion that faculty who broadened their scopes of recognition were able to affirm the science identities of students with fewer incoming cultural resources in science and support their developmental capital. All right, what is our annotation about that? It's that faculty can increase the diversity of science students by affirming previously underappreciated forms of cultural capital, especially affirming capital as it begins to emerge. So this is emphasizing the point that Jen just made through the annotations. And now I'll turn it over to Clark.
Okay, so you may be asking sort of what's the purpose of this study and or this feature that we have. And one of the things that Becca and I look at in collaboration with the editors at LSE is to try to find articles that are very well written and are discussing current and relevant topics. And this paper that we're working with is definitely spot on in that, in all of those categories. And so that was one of the things. Another purpose is to make articles in LSE more accessible to people that might be new to the fields of biology education research. And that was our intended audience. We have been hearing feedback from authors who are using these features as templates for designing their own submissions to LSE. And so that was an unintended consequence, but one we were very happy to see. As Becca just pointed out, in the teaching practices, there are examples in these papers of effective teaching practices, effective research practices, and so we try to highlight those as we go through. As we're annotating, we try really hard to use open source materials. A lot of the references, if you look within the literature, are behind firewalls, and from the making this more inclusive, we really try to get open source resources to the extent that we can. And then as this collaboration points out so vividly, we get to work with authors, and because Becca and I are, we're reading the paper and we make these annotations, and very frequently we have to reach out to the authors and say, are we misrepresenting what you tried to do here? Make sure that we're on the mark. Okay, the next slide, please. Okay, just wanted to note we have 7 articles currently, and variety of subjects. The top one here discusses project ownership and the importance of that in getting students engaged. The second paper is largely on instrument development and how you can design a study and create an instrument that allows you to witness what students are seeing and doing in the classroom. The third one is a great example of both resources in terms of Jeff Shinsky, one of the co-editors in chief now, had put together a great resource, but then he also in this paper takes a look at some of these practices and the impact that it's having on the students. And so that one was also a lot of fun to annotate. The paper that we are discussing today, Recognizing the Potential Among Diverse Undergraduates, illustrates some best practices and also makes faculty very aware of their own biases and some of the things that we can do to try to broaden our ability to include students that the faculty, quite honestly, don't always recognize. And then the other paper is on the roles of use of mixed methods, and this is largely looking at combining qualitative and quantitative methodologies. Again, for people that are new to the field, how to work with qualitative data can be challenging. And this one actually combines both qualitative and quantitative. The science identity, the second to the last one, that is also looking at this idea of developing science identity and how that works to help students recognize their own potential and how well they persist within, in this case, a graduate student population. And then finally, the optimizing efficiency of learning objectives. Use of learning objectives is something that's encouraged, and we very often use that in our classroom. This paper takes a really nice look at what makes the use of learning objectives effective. And so this is a paper that in an annotation that's full of examples of good teaching practices. Okay, and I think the next one is the acknowledgement section. Oh, sorry. The use of, so Becca talked about the lenses. The concept of using lenses was first pioneered in Science in the Classroom, which is a very large scale effort to make articles in the science in the journal Science accessible to students all up and down the education spectrum. And that was it's a very effective tool, and that was where the idea of using lenses came from, was in Science in the Classroom. Okay, now the acknowledgement slide. Okay, so I'd like to acknowledge first, the editors-in-chief. We've had great relationships with Aaron Dolan and now Jeff Shinsky and Kimberly Tanner. One of our key behind-the-scenes people is Justin Peters, who, if you could see the what this looks like when we get done annotating something, and I'm seeing smiles from Jen and Danielle and Becca, it's a mess. And Justin is the person who puts it into this nice format so that it's very usable for you. Okay, Ashante Edwards and Teresa Martin today are keeping things flowing very smoothly. And so they are behind the scenes. We can't even see their faces right now, but thank you for putting this whole program together. We have some NSF support, and then the biology research network that Danielle and Jen work with, and that's the NSF supported part, I believe. Then we have support from the education research group at the University of Georgia. A lot of talent and expertise down there. And then we have some students that have helped along the way: Evan Conway, Zoheb Suleiman, and Jack Cherry. And with that, I think I turn it back over to Cynthia.
Great, thank you all. Becca, will you stop sharing and leave your camera on? Thank you. So first of all, I just want to say that I love the Anatomy of an Education Study feature. I think reading articles that have been annotated in the feature is really interesting. It's kind of like reading with a friend at your side, because you can turn on and turn off the highlights and the annotations. You can read silently without talking to the friend. But then when you want to, you can turn to the friend and you can see what they have to say. What did you think about that? the way they describe the background, the way they describe the experiment. So it's a really engaging way to read the paper. So we're gonna start with a couple of questions about the anatomy of an education study feature and the annotations, and then we're gonna move into questions about the article and its conclusions, okay? So Clark, here, my first question is for you. How do you all decide what to highlight for the background? So for example, in this article, the first 5 phrases you highlight as background are performed, meaningful others, achievement gap, and cultural reproduction of inequality. I guess that's 4, but anyway, first 4 you highlight. So why did you highlight those words as particularly meaningful to annotate?
Okay, so with the background lens, what we are usually choosing to highlight are ideas in the paper where there is some additional information that helps the reader understand it. Very often these will include additional references for further reading. But for us, it's just a chance to say, here is something that you might need to dig a little bit deeper into to fully appreciate. And so that's very often, that's the background lens.
Makes lots of sense. Okay, so I have a related question for Becca. So Becca, I want to highlight one of the words that you annotated with a definition, theoretical orientations. So you define it in this way. In social sciences, theories are logical explanations of social phenomena. Theories explain relationships between variables, which can then be tested empirically. Researchers trained in biology may find it helpful to think of theories as models. So that was the definition y'all gave. Can you talk more about why you chose this word to define, or this phrase in this particular definition?
Yeah, so one of our target audiences is people who are have a similar research trajectory that I have, where we were trained as disciplinary biologists and then got sucked into the world of biology education research. So we don't have the kind of educational research foundation that someone with a PhD in education would have. And theoretical orientation is such a big part of educational research. So we wanted to start explaining what that orientation meant for people who really have the disciplinary biology backgrounds.
And, you know, I love that analogy. You might want to think about it as a model, right?
Yeah.
I want to ask a follow-up question. When I started reading social science literature, I struggled mightily with the idea of theory. How could one have a theoretical lens? What on earth did that mean? Like you, Becca, my traditional training is not in that arena at all. So in addition to your suggestion that biologists may find it helpful to think of theories as models, again, which I think is enormously helpful, I've also found it useful to think about how, as biologists, different techniques allow us to see different things. So I can learn different things about cell structure, for example, depending on whether I use different types of microscopy or use scanning mass spectrometry or use cell fractionation techniques. So the technique I choose focuses me on a particular element of my subject. And I have come to think of theoretical lenses in a similar way. They focus our attention on a particular part of a phenomenon. So Jen, what do you think about that? Like, do you think that this is a legitimate additional way to think about theory, or would you offer modifications?
Yeah, no, I think it's great. I think that actually both Becca and Clark's annotation of the article in some ways helped me understand my own process. I'm going to be honest with you, because there are lots of things that we do, and Danielle and I are both trained as anthropologists, not as education researchers either. So we come actually from strong kind of theoretical and qualitative world. But again, we're sort of new sometimes to the education literature and definitely to the biology world. So in hearing, in looking at this annotation, it helped me learn things too. And I think this analogy of models, but also the analogy of these kind of different techniques, different analytical techniques, are both really good analogies for thinking about it. And even just the word lens sometimes helps me, right? Because if you put a different lens on a camera, you can, zoom in, bring something into focus. But when you bring something into focus, it brings something else out of focus. And I think that that's an important component of this, that whatever it is that we're looking at, we always have to recognize that it helps us understand something from a particular perspective, through a particular lens, right? Through a particular focus. But there are other things that drop away when we do it. And that's sometimes what's useful about having, multiple approaches to the same phenomena. So in our case, we were focused on scientific identity. We were using cultural capital as a lens. But in other places, you could imagine that people are using things like critical race theory and particularly interested in kind of taking that kind of critical race lens for understanding some of these same phenomena.
And I'd like to add, Cynthia, that I'm still struggling with what a theory means from a social scientist perspective. It's my background is in evolutionary biology, and I work with students to understand a very different idea of theory.
Right.
And at conferences, one of my favorite things to do is to be talking to my social scientist colleagues and just exploring this idea of theory. I think it was Tachi Rousseau-Tate who first talked to me about the idea of thinking of theories as models. Mm-hmm. That's really helped. And I think it is because of what you and Jen were talking about, that when you have a qualitative data set, it's so rich in information, and you need to have some lens to focus the analysis. And it's the theory that provides that focus.
Thank you. Well, we have a question from the audience about theoretical grounding. And so Danielle, I'm going to read this to you and ask if you will tackle it. So what approaches do you all use to ensure grounding in a strong theoretical framework?
Well, what we do is we build on the previous literature, and we build on what we've been focusing on in the Biology Research Network for several years, right? So Jen published papers previous to ours on cultural capital, and we wanted to have solid recommendations going to faculty and that we could give to the Biology Research Network to help them ensure that students and their undergraduate research experiences would result in them also developing cultural capital. So we ground it in the experiences that we see with students in the Biology Research Network, but we also, it's what we're experts in as well. So Jen is an expert in cultural capital, but we wanted to provide solid recommendations to faculty, and we knew that we could use cultural capital and that lens in order to develop those for faculty. If we were to use some other sort of lens, that may work too, like Jen said, critical race theory. But with this specific lens, we were able to pinpoint how to help faculty understand how students can develop that cultural capital. So I don't know if that answers directly what, I guess that question has now disappeared, but if there's any other follow-up that you want to know about how we pick our theoretical underpinnings for research, do let us know. A lot of times as social scientists, we pick those before we get into research so that we know what lens we're going to be using. And that's critical because, like Becca said, we have so much information. Hundreds of interviews, and one interview, for example, is 23 pages of text, right? And all of those words become part of the codes that we draw out when we're coding. And so we have to have some sort of lens to look at that, or we're not able to develop those recommendations. Jen, do you want to piggyback on that and give any further information?
Yeah, I just want to say one thing related to that is that, when you're using a theoretical lens and when you're doing qualitative research, not to turn this into a conversation about qualitative research, but how we end up choosing our participants becomes important. And so one of the things that we deliberately do in this kind of work is you try to deliberately or purposefully sample for your interviews in ways that will actually sometimes challenge your theory, because you want to know whether your theory kind of holds up in situations where it could be most challenged. So we wanted to, in this kind of study, overrepresent our students who come from underrepresented groups, but we also wanted to make sure that we had students involved who came from, scientific households with PhD parents, because we wanted to make sure that no matter who we were talking to, or we wanted to be able to also refine our theory based on some of those subtleties. So you deliberately sort of challenge your theory. You don't take it just as face value. And that's one of the ways in which we also ensure that it's that our theory is robust.
And thanks for mentioning that, Jen, because actually one of the things that Clark and I wanted to annotate was how your sampling strategy differs from a quantitative study that a lot of biology education researchers are used to, where you need a random sample. And this is very intentionally not a random sample. And the sampling strategy you used, this non-random sampling strategy, increases the strength of your results. And that's the kind of discordance that we want to be highlighting in the annotations.
Yeah, and I want to ask a follow-up question from the Q&A related to that. So one person says that they're at a small liberal arts college and want to know what words of caution you would have about using qualitative methods to extrapolate conclusions when you start with a small n.
I'll just say something really brief about that, because we could talk all day about qualitative research, but one of the components there is, yes, so purposefully sampling, and there are lots of ways to do that. But in our case, we were looking for a heterogeneity of experience, but overrepresenting, as Becca said, that kind of the students from underrepresented groups. But also the other piece of that is a process called saturation. In other words, we continue to interview and continue to recruit more people until we're not learning anything new, until we walk away from those interviews saying, yep, heard the same thing once again. And one of the differences then again in qualitative research is that that the preliminary analysis actually starts early in data collection, so that you can start refining and wondering if you should be asking your questions in different ways or more deeply. And that's actually another real difference sometimes between qualitative and quantitative work. But that also allows us to know when we've hit the point we're not learning much new. And so we can now be assured that our data set is robust. And usually we throw a few in after that just to be sure about it, but that's what we're going for.
And to build on that, if you're interested in reading some literature on this, Yin has wonderful studies about how to generalize qualitative work and case studies in general. Again, we're able to check with the literature what's going on as well, right? And so that makes it so that our conclusions are more generalizable. And then hopefully you as science educators can look in your classroom and say, oh, I actually see this. I see this happening, and that's another way to kind of check to see if it is generalizable if for your theoretical and for your small sample size.
Thank you. So I want to ask a different question about your sort of choices about how to report from the Q&A. So how did you all decide between using composite profiles versus spotlighting representative students? Did you worry about potential harm from participants recognizing themselves in your paper? that is psychological harm to students who may not potentially have been recognized by faculty?
That's a really good question. We work really hard to make sure that we maintain students' confidentiality. It is true in a small research network like this that there are risks that, especially with a small group of people, that people will recognize themselves or their colleagues. But we try to then not put in so many details that these students couldn't be confused with others, right? But the value really of being able to talk in detail about a student's experience and their history and their trajectory was really critical in a project like this, where the experience that they came to college with, their ambition, and the experiences they were exposed to along the way were actually part and parcel of the analysis. So to create a composite would actually be, we felt like, overstretching our analytical or overstretching our results in ways that we maybe didn't feel comfortable, right? Because we don't feel like any one of these pieces are predictive of a particular outcome.
Mm-hmm.
And so we felt uncomfortable with the idea that we could start making up characteristics of these students. So we felt like that important point really outweighed that risk. But we also did change some characteristics to make it less obvious precisely who the students and who the faculty members were.
And that highlights a really good point in qualitative research. You have to have an institutional review board that has to be approved for human subjects research. And so you'll see in the paper we use names, but those names are not obviously the names of students, those are pseudonyms. And so we have a very strict level of confidentiality that even our partners in the Biology Research Network have no idea where these quotes are coming from. Again, those raw data sets are part of our Jen and I, what we have access to, but no one else has access to those data.
So there's a related question in the Q&A about this. So there's been a push to make quantitative data and analyses public to promote open and reproducible science. What do you think about that, in relation to qualitative data and methods? We need to protect the privacy of human subjects, but you do have really valuable data sets that could be mined in different ways.
So you can upload raw data sets. These need to be completely unidentifiable though. So they could be, part of work that you may know comes from a biology research network. But again, with our human subjects research background, we would not identify the student or the year or anything like that. You can use the words, right? Because that's your data, the raw language and the words from whatever we may be doing, focus groups, interviews, even surveys with open-ended questions, but pinpointing it back to the identifier, we wouldn't upload those.
And I also, I strongly advocate that we don't upload raw interview data. I advocate that if we have colleagues who are doing projects where they want to work with our de-identified data, that we would then collaborate with them and put them on IRBs to allow them access, simply because the trust that comes from interviews and that you build that kind of rapport and that trust is something that is really at risk if you start broadly sharing raw transcripts, unless you start out with that as your plan from the start. So if you're planning your study knowing that this is going to be publicly released but de-identified, and you can tell your participants that from day one, I think it becomes a little bit of a different story because they know that and can make decisions about what to share. But I also think that that can then have a cost in terms of the openness of participants. So I definitely tend to advocate on the not releasing raw transcript avenue.
So now we're gonna pull back out and we're gonna think about that annotation layer on the paper. So we've got a question from the Q&A for both Becca and Clark. Can you all tell us about the aspects of your background, expertise and experience that you have drawn on to help you, A, identify what the audience might need, and B, annotate with appropriate analogies, definitions, and resources?
Okay, I'll start on this and I'm sure Becca can add to it. As Becca related, both of us are converts to biology education research. Our backgrounds are experimental science and very heavy in the quantitative aspects of things. And so part of annotating this is it hasn't been that long where the subject matter was new. And so we, as part of the process, the first go through, we actually over highlight things. And then eventually we end up weeding that down a little bit. But, you know, we're familiar enough with what we knew and didn't know a few years ago that I think that's a good fundamental start. Becca?
Yeah, I totally agree. We use our own ignorance as a guide for what to annotate. And, you know, that has its pros and cons. But to get to another part of your question, what experiences are we drawing on? It's not just our ignorance, it's also our knowledge base. So we're also looking for themes that we see in the biology education research and fundamental works like How People Learn, volumes 1 and 2, right? Those are open access resources that are fundamental in the field. And so we're going to be looking for opportunities to bring those works into the conversation. And we also use our teaching experiences, right? When we're, both Clark and I do some work in professional development, and when we see concepts that are hard for our trainees, we're going to be concentrating on those in our annotations as well. You also asked about maximizing the helpfulness of the annotation strategy. And this is, I would say that this is an area where we're still growing. So one of the purposes of having a webinar like this is to get feedback from you all. Let us know how we can be increasing the helpfulness of these annotations. What else would you like to see? Lastly, I would like to say that we really try hard to make annotations that supplement rather than repeat what the authors have already said. So Clark mentioned that we over annotate in the beginning because we'll be reading and we'll ask questions, and then we find that later on the authors have answered those questions. And this makes areas like the background lens really a little bit tricky because we want to add background information that enriches the conversation rather than repeats the conversation. So with papers that are exceptionally well written, their background sections speak to themselves in some ways, and they just don't. The theoretical framework part of Jen's and Danielle's paper is so strong that we didn't need to annotate it very much because they're answering the questions that readers would have.
I think we're going to end with a question that I am really curious what y'all think about. Okay, so there are a couple of conclusions from the paper that I really want to highlight. So first, students interested in pursuing research science careers were more readily recognized than those interested in medical or health science and non-science careers, especially among faculty who emphasized research in their own science identities. So that was the first conclusion. The second, students with competing family responsibilities struggled to maintain faculty recognition. Okay. Now, I think that there is an unacknowledged conflict that is worth drawing out. So I think that as scientists, we are enculturated to think of the pursuit of new scientific knowledge as an ultimate good. I mean, that's sort of embedded in the idea that we want to welcome more students into science at all, right? And if the pursuit of new scientific knowledge is an ultimate good, then we would particularly want to encourage students who want to and can contribute to that in robust ways. So as scientists, I think that we are implicitly encouraged to think of that big ultimate goal, which has some huge impacts, as you know, I'm waiting to get my vaccine very happily. And we're encouraged to think of that big goal at least as much as the smaller good of helping individuals. So I think this is the source of the differences in recognition that you discovered in your work. Okay, so Jen, I think I'm curious, is there anything in your interviews that touches on this idea?
I mean, I think that you are observing something that's absolutely true. I think this is a really good example of cultural capital. I think this is a really good example of us taking, like, when we are inside a cultural worldview, we oftentimes take for granted as natural and right the perspective that we have on the world. And that doesn't mean that we're bad. It's just the way it is. And I think that our acculturation as researchers is that we get excited about research, and we get excited when we meet students who get excited about research, right? And because we can also identify ourselves as junior scholars with them, and we want to invite them in and we want to support them. We saw this a lot in our research. We saw this especially among faculty who just got really enthusiastic when they met students that reminded them of themselves. And again, I think that this, from a cultural capital kind of perspective, is very natural, but it also has its limitations. And I think one of the things to hold as really important is that students who, for example, were pursuing medical careers, who were the students who were had a very strong science identity, but who were less recognized by faculty than those who were interested in research careers. Those students oftentimes were enthusiastic about medical careers, especially among minoritized groups, as because they viewed those through a lens of a public, a social, a community good. So those students also really viewed that through this kind of ultimate good lens that you kind of propose, but a different ultimate good lens, an ultimate good lens that served their communities and in a very direct way, where it was a little harder for students who did not have science or oriented families to necessarily make the connection, make all of the theoretical and abstract connection from I'm sitting here in a lab, I'm pipetting day in and day out, and this contributes to the ultimate good, because it takes a long time and a number of steps for it to become applied in the real world. And so for a lot of students who didn't have this kind of exposure, that kind of medical careers was a very clear path to contributing to that ultimate good.
Absolutely. Yeah. And I just there's a comment in the Q&A. I want to make it clear, I don't endorse viewing that ultimate good as a reason to only recognize some students. But I think it's a conflict that many scientists probably don't recognize within themselves. And I think it's worthwhile to pull it out so that we can recognize it and then work to recognize all of our students. Okay, so we have 5 minutes left. So Becca, would you show the slides, please? Okay, So we would like for you as participants to type into the chat box. We would love to hear one interesting thing that you learned during today's session. I'm going to do like I do with class, and I'm going to time us. And after 30 more seconds, we'll go on to the next thing. Okay. Becca, would you go on to the last slide, please? And if you're still typing, please continue. We love to hear interesting things that you learned. We would also love to get suggestions for topics or papers to discuss. So please also use the chat box to tell us what you would like to hear in the future, okay? So either for discussions on Online with LSE or papers that you would like to see featured in the Anatomy of an Education Study. Love to see that. And so with that, I want to thank the authors. This was just a really rewarding experience. I think you all did an amazing job telling us about the study and how the annotations help us make the most of the study. And I want to thank Ashanti Edwards, the Director of Professional Development with ASCB, and Teresa Martin as well. So, and all of you, our participants, those are great questions. We have a few left. We will try to address offline. And I really appreciate all of your questions during the session.